We are going to jump now to a Zoom interview with a researcher at MSU. This is all about microbes in Earth's deep soil. I am joined now by our live guest here. Thank you so much for being with us.
Yes, good to be here.
And Jim Teege is who's joining us from MSU. Jim, this is a bit of a complex topic that we're talking about, not one that, everyone might be knowledgeable about. We're talking microbes in deep soil, a new phylum that was discovered through your research. Let's really break down this topic and explain to folks at home, what exactly your research entails.
Okay, so the deep soil environment is a major part of Earth, and it's relatively unexplored. If we think of the deep ocean, it's been explored. We think of the surface soil, it's been explored, but the deep soil environment has not. So we were interested in what microbes live there because we hypothesized that they would be very unique because their environment is so different, particularly because they have very little food. So they've had to adapt and live for a very long period of time with very little food. So that's what we explored and why we did it. And what we found was a completely new phylum. And a phylum is the major group in biology. So phyla are mammals, they're insects, they're reptiles, and we all recognize those as very different because we can see them. But with microbes, they don't look different because they're similarly shaped and they're very tiny, but inside, their makeup is as different as an elephant is from a mosquito. And we find not just one, but we find a whole family of these new types of microbes.
Very interesting. And as far as what a microbe is, I know you're sort of getting into the logistics of this research. What is a microbe? Can you start us there on what exactly that is?
So some people recognize a microbe as a bacterium. That's the most common microbe. And if some of them cause disease, so we often call them pathogens. E. coli is one that people often recognize because it sometimes causes intestinal disease. But most of these microbes are good, not bad. So they do good things for us on Earth.
And that is a huge part of this research, is how these microbes, specifically these ones that you're doing this research on and discovered, have to do with purifying water. Obviously, water is something you and I depend on. Can you walk us through sort of how those two topics intertwine?
Okay, yeah. So what's interesting about these microbes is one is because they're very new and different from what we know already. And there are probably lots of them because this deep soil environment is very broad worldwide. And what do they do? Okay, so the thing that they would do most helpful to humans would be purify water. Because as you know, when water enters the soil, it leaches down through the soil and goes deeper and deeper. And when it gets to these deeper depths, then these microbes can access any pollutants that might be left. So for example, some insecticides or herbicides or gasoline or other types of chemicals like that have gone below the surface layer, it's these microbes then that have the responsibility, I would say, to metabolize those so that our drinking water then is much cleaner.
Gotcha. So yeah, playing a major role, even though these are so small and so deep down, in the earth. And as far as, some people at home might be wondering, I'm wondering, how does it work studying these? Obviously, these are so far under, the ground that we walk on. How does researching something so far away, how does that work? How do you collect specimens?
Okay, so this is a collaborative project, and we did it in two sites, one in Western Iowa, and the other in China. Why China? Because they have very similar to soils to those in Iowa. And in both cases that we can drill very deep. So the Iowa Geological Survey drilled 75 feet deep. That's 3/4 of the length of a football field. And retrieve cores of soil. And then when we get that soil, we take out the center of that core and see what microbes are there. Now, there are some microbes that we can grow in the laboratory on auger plates, but These new ones we can't yet grow. Their physiology and requirements are so different from what we know that we can't grow them. But we can extract their DNA. And then we can sequence that DNA and assemble it to see what is the genetic makeup of these organisms. And we did it both in China and in Iowa, and they are very similar to each other. Not identical, but they are cousins of each other.
And what's sort of, this is a huge discovery. I'm sure a lot of work has gone into this so far. What is next in, this discovery that, you and your team made?
So what is most important now is to be able to culture these in the laboratory so that we can understand how they live in this very starvation environment and for such a very long period of time. These probably have evolved separately from other microbes for 200 million years, for example. So we want to culture them in the laboratory. That's next. Because when we culture them, not only understanding their physiology, but they may have properties that are useful. We often discover new things in microbes, like new enzymes, new drugs, new food products that can be obtained from novel organisms.
Can you touch on that just a little bit more? I think that could be really interesting to people at home watching us right now. This may seem far-fetched from, daily living for most people, but in fact, it's quite connected, as you just mentioned, or at least could be with this new discovery.
Right. Yes. Many of the drugs that we have come from microbes. So people are always looking for new microbes for the possibility that they can produce new drugs. Now, I don't want to overemphasize that because it's still a hard find, but it is a resource that can be used in that manner. It's not only for drugs, but also enzymes, food products, a variety of other things can come from microbes. Because if you can culture them and they produce something useful, you can scale that up in fermenters and it becomes a useful product. So we don't want to promise that, but it is one potential economic use of those. The other, of course, that is certain is that they are metabolizing pollutants that come through the soil system.
Yeah, absolutely. They've got a purpose right now, but could very well develop some additional, uses down the line. But first, that, culturing in a lab, not just way down deep existing in the earth is kind of that next step. I learned so much. I knew this was going to be a complex topic, Jim, but I really appreciate you walking us through it and explaining how this, can impact people who are joining us. So much incredible research happens in so many different topics at MSU. So thank you so much for joining us here on Evening Plus. I really appreciate your time.
Well, thank you very much.
You have a good rest of your night.
Yes.